Thank you, Marlene, for that great talk. So the next talk will be given by Nicholas Turk-Browne. Nick is the director of the Wu Tsai Institute and professor of psychology at Yale University. And Nick's contribution to cognitive neuroscience have been recognized by our own Young Investigator Award, as well as a number of awards from APS, APA, and the like. The title of Nick's talk today is Children Are the Future, the Rebirth of Infant Cognitive Neuroscience.
I read this book in high school by John Maddox, the longtime editor of Nature. In it, he argues something like that the most exciting discoveries in the future will be to answers to questions we don't yet know enough to ask. And so it's a bit presumptuous and likely foolish of me to stand up here and talk about what future questions to ask. But I will tell you a little bit about some of the questions that I'm most excited about. They have to do with this guy. This is my son Calder. This is his first birthday today. It pains me to miss it. We had a long FaceTime this morning, and we'll have a party when we get home. But I've been staying up at night, not just because of him, but also because of several questions about him over the past few years. Just to list out a few of them. What's the nature of the experience of a baby? What do they think about? How do they learn so rapidly? Where do their memories go later in life? What role do I have to play in shaping his mind? What's hidden underneath his somewhat limited behavior? How is he like a machine? How is development akin to selecting a model or training a model? How does the brain self-organize? These are some of the questions that have been asked for centuries by philosophers and decades by developmental psychologists, often using relatively simple and infant-friendly behavioral methods, looking time, sucking rate, reaching and choice behaviors, kicking. And there's been incredible progress in our understanding of infant cognition through these simple measures. On the other hand, these measures are overdetermined. So what a baby looks at and how long they look at it is a function of how salient the stimulus is, attention, how long you show it for, perception, how familiar they are with it, learning, arousal, affect, and many other factors. And so the goal of our work in this area has been to try to look under the hood and to try to determine the different contributions of brain systems to infant cognition and behavior. Perhaps the only thing that rivals the amazing changes that happen in infants' behavior is the dramatic changes that occur in their brains. This is a beautiful study published last year showing dramatic structural changes in the brain over the human lifespan. And you'll see it's a nonlinear scale. A lot of the action happens in the first couple of years of life. Those circles that you see are the inflection point, the rate of maximal change in many aspects of brain structure and anatomy. Now, going beyond anatomy in infants requires the use of some methods that are more infant-friendly to study brain function. Traditionally, these have been electroencephalography, functional near-infrared spectroscopy, and magnetoencephalography. And these have all made important contributions to our understanding of infant brain function. Now, in adult cognitive neuroscience, though, we know that these important tools leave one important tool out of our toolkit, which is functional MRI. And as we all know, functional MRI can provide whole brain coverage and greater spatial precision to complement some of the strengths of these other methods. For example, to be able to study the role of subcortical systems, many of which are fundamental for learning, plasticity, more ventral midline structures of the brain, and to study the cortical surface with great precision. There's plenty of MRI research in infants, for example, while they're sleeping or sedated. Some of this is task-based, using auditory stimuli or other kinds of tasks. But again, in adult cognitive neuroscience, we don't only study sleeping or sedated adults. We're interested in what happens while they're awake and can look at a broader variety of stimuli, engage in more complex cognitive tasks, and link ongoing brain measurements to concordant behaviors. So this is the sweet spot that has driven a lot of the work in my lab and many other labs in cognitive neuroscience over the years. In fact, there are thousands of publications in adults, and yet only a handful in infants when we started working on this. So why? Well, it might be obvious to anybody who does fMRI research why babies are perhaps the worst subjects imaginable. They move a lot, they don't follow instructions, among many other issues. But there were some pioneering early studies. There were 2 ones in that histogram. One of those was from Ghislaine Dehaene-Lambert's in 2002. This was a study of speech perception where infants were played forwards versus backwards speech. And she discovered that the awake infants showed a differential response to forwards versus backwards speech in prefrontal cortex that was absent in the sleeping infants. So this is a proof of concept that it's really important to study infants in an awake state to fully understand the neural correlates of cognition. Since we started working on awake infant task-based fMRI, a couple of other really important studies have come out. One from Maria Cacetta-Morone's group looking at optic flow, so motion processing in the infant brain. And the other from Rebecca Sachs's group looking at category selectivity for faces and scenes in infants. And over the past 6 or 7 years, my group and other groups have been working to increase the throughput and feasibility of task-based awake infant fMRI. I'm going to tell you about some of those projects today to give you a bit of a flavor for what I think might be a path forward in this field. I want to acknowledge up front that this is incredibly difficult team-based effort. I'm going to focus on a couple of studies from Cameron Ellis, who former graduate student, now assistant professor at Stanford, and Tristan Yates, a senior graduate student in the lab. But all of the students and postdocs listed here made very important contributions to the work that I'm going to present. Okay, so how do we do infant fMRI? In our lab, we put baby in the scanner. We wrap them in a vacuum pillow. This doesn't restrain them, but it helps to prevent them from rolling around or rolling off the table. We use a lot of hearing protection, and this is, of course, to protect the hearing, but also to avoid startle responses for loud scanner noises. That's a video, in fact, on the right of a little baby during a scan, reaching up towards a camera in there. The father's rubbing his chest. As you'll see on the top of the ceiling of the scanner bore, there's both a screen and a little red box there. That red box is to indicate an eye tracker, and so this is essentially a MRI-safe webcam. And with that, we can record eye movements coarsely and manually code them to figure out whether a baby's engaged in the task and where they're looking. You might also notice that the baby's head is protruding out of the head coil, and that's because we're only using the bottom of the head coil. So for aficionados, this might seem a bit odd, but we validated in early work that because of the smaller size of the infant head and brain, we get good SNR over their whole brain with just the bottom elements of the head coil. This is a critical decision because it allows the parent who's in the room and the experimenter to readily see the baby in their face, make sure they're comfortable. It allows us to present visual stimuli on the ceiling of the scanner bore, as you see with that screen. And also for safety reasons, with older infants, sometimes they just sit up or crawl out of the scanner during a scan, and this allows us to, not make sure that they don't hit their head when they do that. The screen is projected from behind on a mirror that reflects onto the ceiling of the scanner bore. Pretty wide field stimuli, more than 40 degrees of visual angle. And this display is right over the baby's face. We discovered early on they don't love looking in mirrors, and so this allows for a very immersive kind of task design. In addition, as I mentioned, there's a parent in the room, and the guy on the other side of the table is me. This is Cameron's attempt to render me as a model. This kind of surfer dude model in our VR set. But one of the experimenters is always in the room as well. And in fact, I've gone to most of these scans myself, and it's been a real joy to participate in this data collection with my students and postdocs. I'm not going to go into too much detail, but it turns out that a lot of the standard methods that we use for task design, for preprocessing, for data analysis don't work out of the box with infants. And so we've spent a lot of time developing new methods for seamlessly running tasks and doing analysis. To give you a bit of a summary of where we're at, this is a graph that shows in each column one session of data collection. So this is 260 or so sessions. What's on the x-axis is the age of the infant at the time of the session, and on the y-axis is the amount of data collected in minutes. To orient your attention a little bit here, if you focus on the dark green, that's the amount of functional data we collected that's usable. And so what we're talking about is 5 to 10 minutes of usable data in young infants. Again, for adult cognitive neuroscience, this would be dreadful, but it's the best we can do. You'll see there's also an age effect. We get more data in older infants. But there's also some infants who are young who produce a lot of data. My student Lily has been doing some interesting analyses over all of our studies to try to figure out what predicts whether we're going to be able to collect a lot of data. Age is a big factor. Turns out sex is a factor. And other interesting things, like we have parents tell us how much screen time the baby has, and it turns out babies who don't see a lot of screens produce more data. We can also look at factors of the experiment. So for example, we had the intuition that if we brought babies back for a second, third, or fourth session that they would do better. They'd be more familiar with the environment. Not true. The experiment type does matter, though. We get more data from naturalistic movie-based experiments or block designs than we do from event-related studies. As another fun variable, time of day matters as well. So you get more data at late afternoon and mid-morning. So across these studies, we've been trying to conduct some proofs of concept that the data that we collect can replicate some fundamental functional properties of the brain, and also now begin extending it into studying learning and memory in infants. So I'm going to quickly just mention a couple of studies that are early on that helped us believe in the data that we were collecting. So this is some of Cameron's work performing retinotopic mapping in individual infants. So this is a 5.5-month-old infant from whom we could define the boundaries between areas V1 through V4. This is an attentional cueing paradigm, a Posner task, essentially, where we measure overt attention to a target as a function of the validity of a cue, and we're able to both replicate behavioral effects in the scanner and interestingly find that many of the key regions in adult attention networks are engaged in infants reorienting to an invalid target, including the anterior cingulate cortex and the frontal eye fields, or what look like them. Tristan's conducted several interesting studies, one on event segmentation, and so many of you would be familiar with the work by Jeff Zox and others showing that adults consistently segment events and that we experience continuous inputs in discrete chunks. You can't ask an infant to parse a sequence overtly, and so instead we use data-driven methods to look at state changes in their brain, and from this able to recover very reliable boundaries between continuous sequences. Interestingly, infants, even in early sensory regions, parse at a coarser or longer time scale than adults. She's also conducted an interesting study on face perception. This was somewhat of an accidental study. We were looking at facial identity processing in the infant brain, and then we stopped for COVID. And when we came back to complete the sample, we discovered that the babies that were collected before the COVID lockdown and after, who were born during the lockdown and had the first few months of experience during that, looked very different. So here what I'm plotting is when the identity is repeated versus novel, so this is a repetition suppression kind of design. The pre-lockdown babies showed repetition suppression effect, whereas the post-lockdown babies showed repetition enhancement. Okay, in my last 10 minutes, I'm going to give you a bit of a story about an ongoing and I think future direction to try to understand the principles of how infants learn and remember. In my lab in adults, we've been working for a long time on the relationship between episodic memory, our memory for discrete moments in our lives, autobiographical events, such as this restaurant I went to, last time I was in San Francisco, who I was with, what I was eating, what it smelled like. This is a garlic restaurant for those who don't know. And then also, in contrast to this sort of one-shot discrete memory, our ability to pick up on regularities across experiences. So for example, to learn how restaurants look, or the sequence of events when I go to a restaurant and sit down, I'm given a menu, order, eat, pay, etc. Or the map of a city and which neighborhoods tend to have good restaurants in it. All of these kinds of knowledge require extracting regularities across multiple experiences. In traditional theories of memory, these are fundamentally different processes with opposing computational requirements. So for example, complementary learning systems theory says that the need to form one-shot memories in episodic memory very rapidly and to store those memories separately from each other to avoid interference is fundamentally at odds with our ability to learn more gradually across multiple experiences and to store memories in an overlapping way in order to extract their commonalities. The solution in standard adult theories is that these functions must therefore be supported by different brain systems. My former graduate student and one of this year's winners of the Young Investigator Award, Anna Shapiro, proposed how these two kinds of learning and memory might in fact be mediated by the same brain system in the hippocampus. Instead of only depending on separate hippocampal and cortical systems, she showed in a really elegant computational modeling paper that episodic memory is supported by one pathway in the hippocampus from entorinocortex to dentate gyrus to CA3 to CA1, called the trisynaptic pathway. And the statistical learning, or this more integrative kind of learning, is supported along the monosynaptic pathway that projects from entorinocortex to CA1. This helps to explain potentially a lot of fMRI and neuropsychological and intracranial findings of hippocampal involvement in statistical learning. So this was the background that led us at first to study infants, in fact, which is a bit of a mystery. Infants are remarkably good statistical learners. In fact, the first statistical learning paper was from Jenny Safran and Dick Aslan and Alyssa Newport in 8-month-old infants. There have been subsequent studies by Scott Johnson and others showing that even newborn infants are really good at statistical learning. And our work in adults suggested that this ability depends on the hippocampus. But in many ways, the field of memory considers the hippocampus immature in infancy because its important role in forming episodic memories doesn't seem to come online until a few years old, when we're able to first lay down our sort of durable episodic memories, and that this is the root of infantile amnesia, the fact that we can't remember anything from the first few years of life. So if you go back to the model, our hypothesis is then that maybe these two pathways in the hippocampus allow the hippocampus to support statistical learning earlier in development than it's able to support durable episodic memory. There's some support for this, circumstantial support for this idea in, if you look at the anatomy of the developing macaque. So this is an infant macaque's hippocampal circuit. The monosynaptic pathway emerges in a juvenile age, whereas the trisynaptic pathway doesn't fully emerge until adult macaque. And so this differential developmental trajectory of these pathways suggests that we may find evidence for earlier statistical learning than episodic memory in the human infant hippocampus. So I'm going to briefly show you two studies, one on statistical learning in infants, human infants, and the other on a rudimentary form of episodic memory. The statistical learning task looks like this. Babies were shown a continuous sequence of looming fractal patterns, one after another. They were presented in blocks, so past is prologued in. We're back to block designs, but remember that we're talking a few minutes of data here. And we're going to compare hippocampal activation to these blocks in which there was a reliable pair structure. So item A was reliably followed by item B. What followed item B was the beginning of another pair. To blocks in which a different set of fractals was presented in a random order, where there were uniform transition probabilities. Based on the adult literature, we hypothesized that we'd see greater hippocampal activation for the structured blocks than the random blocks as infants learned a sequence, if the hippocampus is involved in early statistical learning. What I'm going to plot here is a summary of activation in the hippocampus. We manually trace the hippocampus in a set of 24 infants. And I'm going to plot the difference between structure and random blocks over time. Again, a small amount of data. We're going to look at this crudely, the first versus second half of exposure. And indeed, what we see is that although there's no difference between the structure and random blocks in the first half of exposure, by the second half it emerges. This effect is not correlated with age. It seems to be slightly stronger in the right hemisphere, and in fact, this is true in adults as well. And if anything, a bit stronger in the anterior part of the hippocampus. In adults, the CA1 subfield, that's the core of the monosynaptic pathway, is overrepresented in anterior versus posterior hippocampus. So this is consistent with this pathway idea. Okay, so we see some evidence of statistical learning in the infant hippocampus in humans, infants as young as 4 months old. What about episodic memory? Again, past as prologue, here we're going to use the same kind of subsequent memory design that Dan referred to, but made a baby-friendly version. This is psychedelic pattern is to keep their attention. As I showed you before, babies really do not like event-related designs, and so we have to distract them during gaps. And so they're shown discrete images like these scenes, objects, and faces. And after variable delay and several intervening items, we test their memory using visual paired comparison. So again, we're measuring their looking behavior in the scanner. We're going to present a pair of images, one that's old and one that's new, and see what they look more at. Generally, in this kind of a design where you do one-shot exposure to a picture, infants tend to look longer at the familiar item. They're not being fully habituated to that item, so they tend to show a familiarity preference. So we can use this measure of subsequent familiarity as a way to post hoc code the encoding trials and ask what was happening during encoding for pictures that they later showed a familiarity preference for. So here's an example of a baby. Just to give you a sense of the eye tracking data, test trial is going to come up in a second. And you'll see him look left and right, and we're going to manually code whether he's looking more at the familiar or novel thing. Again, a summary analysis in this case, again, measuring hippocampal activation from the hippocampus. First, I'm going to collapse across all infants and show you that indeed, during encoding of pictures that later lead to a familiarity preference, we see greater activity. So here we see early hippocampal evidence of something akin to item memory or episodic memory for individual items that leads to this later familiarity preference. Now, I showed you with statistical learning this did not depend on age, but in the case of subsequent memory, we do see a very strong age effect where it's driven entirely by older infants. So if you do a median split on age, it's infants over a year old whose hippocampal activity during encoding predicts later looking. In fact, there's a continuous relationship here too. To summarize this part, we found evidence for early involvement of the human hippocampus and in exploratory analysis, the medial prefrontal cortex, in statistical learning across infancy. Slightly later engagement of the human hippocampus and parahippocampal cortex in exploratory analyses during visual encoding that drives later subsequent looking at those same items. And what we're working on right now, and what I think is promising for the future, is to try to study longer term, more remote aspects of episodic memory. Here we're talking about delays of 30 to 90 seconds within session, but infantile amnesia is obviously something that can extend for many years, our entire life. And so what we're doing is collecting home videos from babies. This is an example, not from our study, but from the SACAM data set of a father taking his baby down a slide. So we collect these home videos of sort of sometimes mundane, sometimes more exciting events at home. And then we can use these as stimuli in later scans to test memory for what the baby encountered early in life. So we can scan babies at different intervals from when the videos were collected and at different ages to look at forgetting curves over infancy. Here, this was a pilot study we ran where we recruited 2 of the kids who participated in the SACAM study to come back and do a scan with us. And what we found is that the 3-year-old who came back to watch videos from when they were an infant showed greater hippocampal activity when watching videos of their own experience than when watching videos of another kid's experience. But by 9 years old, this effect, this response to their own videos, where they might have some memory, was gone in a different kid. And so we're doing this longitudinally with the bigger sample. Okay, so where does this leave us? I think the main take-home message I wanted to share today is that awake, task-based infant fMRI is feasible. If you're interested, we're happy to share and help. I've put a link here to all of our papers, and perhaps more importantly, all the data that I've shown is public, and all the software used for tasks and data analysis is also available. Perhaps more than all these technical pieces is it really requires an incredible team of really generous and selfless researchers who contribute to each other's projects, and I'm very fortunate to work with many incredible students and postdocs. And stepping back one more layer, I just wanted to say that even if you're not interested in babies or memories, baby or learning or memory, that to me, a lot of the future of the field lies in interdisciplinary research. We heard about some of that in Dan's presentation and Marlene's presentation, trying to bring to bear our understanding of molecular, cellular, developmental systems biology, on behavior, and bringing in ideas from computational fields, applied math, physics, computer science, data science. It's often these sort of collisions and collaborations between fields that lead to really revolutionary progress. I'm part of one initiative like that at Yale University. You can check out our website if you're interested for fellowships and other opportunities, including in the Center for Neurocognition and Behavior, which is one of our research pillars that'll be directed by Kia Nobre, and we'll have a really nice facility for invasive and non-invasive human neuroscience, including several new technologies. And so I'll stop there, and thank you very much.